In today's video, we will be breaking down the Watson and Crick model of DNA. We will be exploring the different structures inside DNA and their bonds that work together to create the DNA double helix model. The DNA strand is made up of nucleotides all connected together. The structure of a nucleotide includes a phosphate group, a deoxyribose sugar with 5 carbons, and a nitrogenous base. Each carbon of the deoxyribose sugar has a specific function. The 1' carbon attaches to the nitrogenous base, the 2' carbon distinguishes DNA from RNA, the 5' carbon bonds to the phosphate group, and the 3' carbon bonds to the phosphate group of the next nucleotide. The image below labels the deoxyribose sugar with these carbons and shows the structure of a whole nucleotide. There are 4 nitrogenous bases found in DNA, and they're broken into 2 groups, purines and pyrimidines. Purines have a double ring structure and consist of adenine and guanine. Pyrimidines have a single ring structure and consist of cytosine and thymine. Uracil is found in RNA instead of thymine. Adenine and thymine pair together using 2 hydrogen bonds, while cytosine and guanine pair together using 3 hydrogen bonds. Hydrogen bonds are weak, though, and are not the main stabilizing force of the double helix structure. Base pairing between 2 strands of nucleotides allows for a constant width in the helical structure and agrees with Chargaff's base pairing rules. This rule states there will be an equal number of A and T bases and an equal number of C and G bases. Now that we have looked at the structure and bonds included in base pairing, we can zoom out and look at the structure and bonds involved in the creation of the double-stranded DNA. Phosphodiester bonds connect the sugar-phosphate backbone. The 3' carbon of one deoxyribose sugar bonds to the phosphate group on the 5' carbon of the next deoxyribose sugar. This will give you the 3' to 5' direction of the DNA strand. One property of DNA is the fact that it is anti-parallel, meaning that one strand will run in the 3' to 5' direction and the other will run in the opposite direction, 5' to 3'. This can be seen in the image below. After discussing the double-stranded structure of DNA, we can move on to the double helix structure that these strands form. The main force in stabilizing the double helix is base stacking, which maximizes the hydrophobic reactions of the strands. The hydrophilic backbone is on the outside, while the hydrophobic bases remain in the center. DNA can be right-handed or left-handed, but what Watson and Crick discovered is that, and what most of the scientific community follows, is the right-handed model. This means the helix coils up and to the right, showing 10 base pairs per turn. The helix also has a major and minor grooves shown in the image to the right. These grooves expose the edges of each base pair so that proteins can chemically recognize the DNA sequence. The phosphate backbone of DNA has negative charges present, which can destabilize the double-stranded DNA. If we were to put DNA into a salt buffer, such as saline sodium citrate, the salt ions would partially neutralize these negative charges, stabilizing the double helix structure. Tm stands for midpoint of thermal denaturation, when 50% of the DNA is denatured. By putting DNA into a salt buffer, the midpoint for thermal denaturation would increase as salt concentration increases. These are my references. Thank you.